Something about this sample doesn't seem right. Compared to healthy tissue, many aspects of this sample indicate uncontrolled abnormal cell proliferation, in other words, cancer. Normally, epidermal growth factor, or EGF, initiates a signal for cell proliferation by binding to the EGF receptor on the cell surface. In turn, a critical protein called RAS is activated. Let's take a closer look at RAS. When RAS is bound to GDP, it's inactivated. The binding of EGF to EGFR triggers a guanine exchange factor to release GDP from RAS. GTP then binds to and activates RAS, with the extra phosphate holding two switch regions in a loaded spring configuration. Activated RAS sends a signal from the EGFR through the RAS-RAF-MEK-ERK pathway. At the end, the signal is relayed to transcription factors in the nucleus. This process eventually results in the production of proteins that are essential for cell division and other processes. To prevent cells from dividing nonstop, the RAS protein inactivates by cleaving off a phosphate from GTP after relaying the signal to the next protein in the pathway. The hydrolysis reaction is accelerated by the arginine finger of a GTPase activating protein. This finger interacts with the alpha carbon of the G12 residue in the RAS active site to stabilize the transition state. A G12 mutation in the RAS gene will replace glycine with a bulkier amino acid and interfere with this interaction. Thus, hydrolysis is greatly slowed. The carbonyl group of the arginine finger also interacts with the side chain amide of the glutamine 61 RAS residue. The side chain carbonyl of this glutamine properly positions a water molecule for hydrolysis. If this residue is mutated, the RAS protein's intrinsic GTPase activity will be greatly reduced. So what's the end result? By preventing RAS from hydrolyzing GTP, the structural changes brought about by RAS gene mutations permanently activate the protein. It'll continuously signal cell proliferation, which may lead to tumors, cancer, and metastasis. Each year, over 1.6 million patients are diagnosed with cancer in the US alone, and RAS mutations are associated with over 30% of all human cancers. Greater knowledge about these mutations open up new opportunities for cancer treatment. While we wait for a cure, we should practice a healthy lifestyle, avoid carcinogens, watch out for cancer symptoms, and go for routine screenings. Measures such as these all serve to prevent the devastating effects of oncogenic mutations.